This is a Biology IGCSE Edexcel 1B paper from 2023. The diagram shows a flower from an apple tree with some structures labelled. This apple tree is able to self-pollinate, which is the correct transfer of pollen during pollination. So remember, pollen is made on the anther and then it gets transferred to the stigma in pollination. So we need to start with an anther here. We need to transfer it to a stigma, so it's V to P, which is C. Which structure does the pollen tube grow down? We're looking for the style, which sits below the stigma, which is Q, A. Which structure develops into the seed? We need to locate the ovule, which is T. The flower from an apple tree is insect pollinated. Give 3 differences between the structure of this apple flower and the structure of a wind pollinated flower such as grass. So we'll start by saying that the apple flower has large flag-like petals. Remember, that's to attract the insect. The apple flower has colourful petals, again, to attract the insect. And then we need to say that the apple flower has enclosed stigma compared with the wind-pollinated one, which has exposed stigma. The seeds of many plants are surrounded by sweet-tasting fruit. Suggest how this enables the plants to spread their seeds. So that fruit is therefore highly attractive to animals, and therefore the animals swallow the seeds, and then the seed is dispersed when the animal poops or egests feces. The diagram shows a transverse section through a human heart. Draw an X on the diagram to show the position of the septum. So the septum is the division between the left and right hand sides of the heart, hence why we want to put our X here. State 2 differences between the composition of blood in the coronary artery and composition of blood in the coronary vein. So the artery, remember, contains oxygen or more oxygen. And then the vein contains more carbon dioxide because remember, the cells of the heart will have been respiring. Explain the differences between the left ventricle wall and the right ventricle wall. Look, it's much thicker on the left side compared with the right side. So why is that? Because it contains more muscle. And it needs more muscle so it can contract at higher pressure. And it needs to contract at higher pressure to force the blood all around the body, as opposed to the right ventricle wall, which just forces blood to the lungs, which is much closer. The development of coronary heart disease is linked to a number of factors. Explain how these factors can increase the risk of developing coronary heart disease. So, factors include smoking. What does that do? Well, it increases the viscosity of the blood, which makes the blood thicker, meaning that blood is at higher pressure. And high pressure is bad because it encourages coronary heart disease. We could also say that a lack of exercise means blood pressure isn't lowered. Diets high in saturated fat. May lead to fatty deposits in the coronary artery. Stress also increases blood pressure. So we've said 4 really good points here. The diagram shows the nutritional content of 2 non-dairy milk products, oat milk and almond milk. A person is told by their doctor they need to lose weight. Use the information from the milk contents and your own knowledge to discuss which milk would be most suitable. So just have a look. It's going to be almond milk in terms of losing weight. So almond milk provides less energy and is therefore better suited to someone trying to lose weight. Now we'll just go through the other nutritional points. So almond milk contains less fat. Almond milk contains more sodium. Almond milk contains less carbohydrate. Just really writing out what this data is telling us. Almond milk contains less fiber, so the person may suffer more from constipation. And then I'm finishing off by mentioning protein. Almond milk contains less protein. Remember that protein is responsible for muscle growth and repair. Okay, I've said loads there. Suggest why a person might drink a non-dairy milk, such as oat or almond milk, rather than cow's milk. Lots of options here. They could be vegan. You could also say that they could be lactose intolerant. Describe how a student could test a milk sample for glucose. Add Benedict's reagent. Make sure you have these food tests sorted. Heat. And it should turn brick red. That's your positive result for glucose. Human breast milk contains special proteins that give immunity to the baby. Explain how these proteins can help protect the baby from disease. So the proteins are antibodies. So the antibodies kill pathogens or bacteria. And what do they actually do to the pathogen? They help stick the pathogen together, making it easy here for phagocytes to engulf them. The peppered moth is found in many countries. 2 different forms of the peppered moth are the light-coloured moth, dark-coloured moth. The dark-coloured moth was first observed in cities when pollution from burning coal stained tree trunks black. So therefore we know that this black moth is better camouflaged in places where there's lots of pollution. Scientists trapped moths in a city location from 1992 to 98. The table shows the scientists' results. Calculate the difference between the percentage of moths that are dark coloured in 1992 and the percentage of moths that are dark coloured in 1998. So we're looking at these two bits of data. So we'll do 27 divided by the total, which was 36, times by 100. So that's 75%, versus in 1998, when you have 9, so that's 41%, and then we work out the difference between these two numbers, which is 34%. Plot a line graph to show the number of light-coloured moths and dark-coloured moths from 1992 to 1998. Use a ruler to join the points with straight lines. So let's steal the data. Remember, what you measure, the dependent variable goes on your y-axis. So that's the number of moths. What you change, your independent variable goes on the x-axis. And then let's pick a sensible scale from 1 to 27. 5, 10, 15, 20, 25, 30. That's good. So let's go for light-coloured moths. 1992, and it was 9, 11, 5, 1, 8, 13, and it said use straight lines to join the points. And I'm going to need a key. So that's the light-coloured moth. Now let's do the dark-coloured moth. So we start at 27, 18, 7, 1, 4, 1, and then lastly 9. Join these up with straight lines. So that's our dark-coloured moth. So I'm happy I've got 5 marks there. In the 1990s, many cities introduced laws that prevented the burning of coal. Comment on the changes in the number of light-coloured moths and the number of dark-coloured moths between 1992 and 1998. So we need to start by talking about what's going on in terms of describing the graph, and then we'll talk about reasons for that. That's what that comment means. So if we start with the black line for the light moths, we can see that the number of light moths increases, then decreases, then increases, whereas the number of dark moths, first of all, decreases and then increases. So why would the number of dark moths decrease? Let's think about what's happening in terms of pollution, because less coal burning means fewer dark buildings. So black moth is less camouflaged. More predation, so it gets eaten more. As it's more easily seen. So in terms of the marking points, there's a mark for talking about describing what's happening to the dark and light moths. We talk about the dark moths decreasing in number because there's less coal burning. 1 mark, meaning that they're less well camouflaged, is a mark. More predation occurs because they're more easily seen. So we've absolutely got our 5 marks there. The chromosomes in a human cell can be photographed and then arranged in pairs to show the karyotype. White blood cells are often used to show the chromosomes in the karyotype. The chromosomes in white blood cells are larger and easier to see when the white blood cells divide. Diagram 1 shows the karyotype explain why a red blood cell cannot be used to show a karyotype. So remember, chromosomes are found in a nucleus and red blood cells don't have a nucleus. So totally useless for finding a karyotype. State the type of cell division that occurs in white blood cells. Well, you're just trying to reproduce these white blood cells, so we're going to be using mitosis. The karyotype in diagram 1 is from a male. State how this can be deduced from the diagram. So we are looking here at the sex chromosomes, and we're expecting, as we can see here, to see an X and a Y chromosome. Diagram 2 shows the karyotype from a white blood cell from another person. The karyotype is from a female, and the person has a condition called Turner syndrome. The condition affects the development of the ovaries, so they may not produce the normal quantities of sex hormones. Oh yeah, look down here. Comment on the differences between the karyotype shown in diagram 1 and diagram 2 and the effects of Turner syndrome will have on the person. Use information from the question and your own knowledge in the answer. So first of all, we can see a missing X chromosome. So diagram 2 is missing an X chromosome. So what's the issue here? Well, that means that this person might not necessarily be able to undergo the usual changes at puberty because they can't release estrogen, and therefore they don't develop secondary sexual characteristics. Also, that lack of estrogen means they might not be able to repair their uterus lining. Suggests how the difference in the chromosomes of people with Turner syndrome may have been produced. Often when you see a change in the chromosomes like that, it's going to be due to mutation, so a random change in DNA. A student uses this method to investigate osmosis in potato tissue. Cut 3 5cm long cylinders from a raw potato. Dry the cut surfaces using filter paper. Measure the mass of each cylinder using a balance. Place one cylinder in a test tube containing 10 centimeters cubed of concentrated sucrose solution, one cylinder in a test tube containing 10 centimeters cubed of distilled water, and one cylinder in an empty test tube. Place a bung in each test tube and leave them for 1 hour. Remove the cylinders, dry them, measure the mass of each cylinder again, measure their lengths using a ruler. Okay, so I can already see my independent variable is the solution. That's what I've changed each time. Describe what is meant by the term osmosis. It's the net movement of water from an area of high water potential, so somewhere where there's lots of water, to an area of low water potential, across a semi-permeable membrane or partially permeable membrane. State the independent variable. I've already said that. It's what we're changing, the concentration of solution. Osmosis is affected by the surface area and volume of cylinders, so the student keeps the original surface area and volume of each cylinder the same. At the start of the experiment, each cylinder had a radius of 0.25 centimeters and a length of 5. Calculate the surface area of each cylinder using this formula. Okay, so plug your numbers in nice and carefully. So we're doing 2 pi, they've told us that pi is 3.14, times the radius, which I've been told is 0.25, times the length, which is 5. So let's do that as one calculation, plus 2 times pi again, that's 3.14, times 0.25 squared. So I was just dealing with the second portion of that. Put that into your calculator nice and carefully. So you'll get 7.85 for the answer to that first bit of the calculation, plus 0.3925. And then your final answer is going to be 8.2 to 2 significant figures. Explain how surface area would affect the rate of osmosis. So higher the surface area, the faster the rate of osmosis. Why is that? We're being asked to explain after all, because there's more contact. Between the potato and solution. State another variable the student should control. That means keep the same. So think about the factors that will affect the rate of osmosis. Temperature is a good option here. You could have also said the type of potato. The table shows the student's results. Explain the changes in the mass of the potato cylinder in each test tube. So we'll start here with concentrated sucrose solution. Remember that sucrose solution will have a lower water potential compared with the potato, which means that the potato chip will lose water. And as you can see, that's seen here with a minus 0.3 grams. So in the sucrose solution, water leaves the potato by osmosis from an area of high water potential, that means water potential, in the potato to an area of low water potential in the surrounding solution. Hence, the potato loses mass. So we've dealt with that one. What about in distilled water? Well, there'll be a high water potential in the surrounding solution, the distilled water, so water enters the potato, as we can see. So in distilled water, water enters the potato, and then in air, let's have a look. There should be barely any change. Yeah, no movement of water occurs. That's really acting as a control. The student also measured the change in length of each potato cylinder. Assuming the length has the same percentage change as the mass, calculate the final length of cylinder in the concentrated sucrose solution. So let's scroll up and look at the data, and we want to first of all calculate the percentage change in mass. So we take that change in mass minus 0.3, we divide it by the original mass, times it by 100 to find a percentage decrease in mass of minus 14.3%. And then in terms of finding the final length, we know that the initial length is 5 centimetres. So effectively the final length will be 85.7% of that initial length. So we just do 5 multiplied by to get an answer of 4.3 centimetres. This food web is from an ecosystem in Africa. Which organism is the primary consumer? So primary consumer feeds on a producer. which is something which photosynthesizes. So we're looking for anything that's coming straight up from our producer. So what do we have here? I can see that we have a zebra. Which of these has the least efficient energy transfer? Now, the least efficient energy transfers occurs from the producer to the primary consumer level, which is why there's the only producer mentioned, so it'll be stargrass to baboon. Which organism will be least affected by a reduction in the population of stargrass? So in order to answer this, let's have a look through the options: baboon, gazelle, wildebeest and zebra. Baboon's definitely going to be affected because its food sources decreased, as has the zebra and the wildebeests. So therefore, let's have a look. The gazelle also feeds on the acacia, so that will be okay because it's still got acacia to eat. So the answer here is gazelle. Explain why only a small proportion of the energy contained within one trophic level is transferred to the next trophic level. Gosh, this comes up so often. You must learn this. It's because not all of the organism is eaten. Some of the organism is indigestible. There are losses in respiration, heat loss, movement. Just be prepared to list this: egestion, which remember is pooping, and excretion, which is removal of things like urea, so metabolic waste. Describe how scientists can compare the population size of stargrass in 2 areas of an ecosystem. I'm hoping this is screaming quadrats. So use quadrats, make sure you spell this word properly, which have been placed randomly. Then your next step is to count the number of stargrass and then multiply up your numbers. To gain an estimate for both areas. And always repeat and calculate an average. Wild dogs hunt a variety of prey species. They usually eat the weak and sick. Explain how this behaviour may benefit the species the dogs hunt. Oh wow, that's survival of the fittest. Wow, okay, so this is actually Darwin's theory of natural selection. So it means that weakest prey are eaten, meaning they're less likely to survive and reproduce. So they're less likely to pass on their sickly alleles. You can obviously argue it the other way around if you want to talk about it's more likely that the stronger ones survive. And then just generally, by removing the sick animals, yeast can be used in food production. Which group of organism does yeast belong to? You must learn that. It's a type of fungi. Which substance is in the cell wall of a yeast? It is chitin. Cellulose, remember, is in plant cell walls. A teacher sets up an experiment to investigate the effect of temperature on the rate of respiration in yeast. The diagram shows part of the apparatus the teacher uses. Explain what additional apparatus the teacher will need to investigate the effect of temperature on the rate of respiration. Okay, well, we need a way of measuring the temperature, so we need a thermometer. We need a way of controlling the temperature, so a hot water bath. And then lastly, it's this point about the rate of respiration. Rate always indicates time, so you need a stop clock. State the purpose of the liquid paraffin on the surface of the glucose solution. So that liquid paraffin will stop oxygen entry and therefore force the yeast to anaerobically respire. Give the name of a suitable chemical that could be used as an indicator X to show that the yeast is respiring. So we know that we need to have a way of detecting carbon dioxide, so you could use lime water or hydrogen carbonate indicator, whatever you prefer. I'm going to go for lime water. Diazine green changes colour from blue to pink as the oxygen levels in the yeast and glucose solution reduce. Explain how this colour change gives information about the respiration in yeast. So the reason it's blue to begin with is because initially that yeast is respiring aerobically, so using oxygen, and then it changes to pink as the yeast begins to anaerobically respire because all that oxygen has been used up. Explain why the rate of respiration in the yeast will change as the temperature is increased. So as soon as you're talking about temperature, you need to be talking about enzymes. So let's start by saying initially the respiration rate increases with increasing temperature because the enzymes have more kinetic energy and form more enzyme substrate complexes. Once you reach that optimum temperature and you increase the temperature further, you're going to find that the enzymes denature, meaning that the respiration levels decrease. So as temperature increases, enzymes have more kinetic energy, more enzyme substrate complexes are formed. An optimum temperature is reached. Above the optimum temperature, the enzyme denatures and active site changes shape. Plants need light to photosynthesize. Give the balanced chemical equation for photosynthesis. So we don't want words, we want symbols. So plants use up carbon dioxide and water. They produce oxygen and glucose. And then remember, you need sixes to balance that. It's worth learning that equation off by heart. The graph shows the effect of light intensity on the rate of photosynthesis in a water plant. The rate of photosynthesis is measured by counting the number of bubbles of gas released per minute. The light intensity is decreased by moving a lamp further away from the water plant. The light intensity is calculated as, using information from the graph, calculate the distance of the lamp from the plant when the rate of photosynthesis is 78 bubbles per minute. So we're going to steal this equation, write it out again in the exam. So we know that's the light intensity. So let's find the light intensity when there's 78 bubbles per minute. So read across here, read down here, and you can see that that's 0.16 equals 1 divided by the distance in centimetres of the lamp. So that's what I'm trying to solve. So cross multiply to do 1 over 0.16 and then square root that final answer to find x, which is 2.5. Describe the relationship between the number of bubbles per minute and the light intensity. So as you can see here, as light intensity increases, we can see the number of bubbles released increases. And this is a very steep relationship initially, and then the number of bubbles levels off. Explain the rate of photosynthesis between a light intensity of 0.4 arbitrary units and a light intensity of 0.8 arbitrary units. We're looking here. Okay, because increasing that light intensity has no effect on the number of oxygen bubbles released, you can see that because it's plateaued, it means there must be another limiting factor. So that could be temperature or it could be carbon dioxide concentration. So between 0.4 and 0.8 arbitrary units, there's no increase in the number of oxygen bubbles, meaning there's a different limiting factor. And then name an example of an alternative limiting factor. So that could be temperature is too low, carbon dioxide levels are too low. The table gives some information about enzymes in the human digestive system. Complete the table by giving missing information. So the enzyme which digests starch is known as amylase. Maltase breaks down maltose to form the reducing sugar glucose. Protease acts on proteins, and lipids are broken down into fatty acids and glycerol by lipase. So do get those digestive enzymes learnt. Some scientists have investigated the effect of vinegar, weak acid on the digestion of starch. Design an investigation to discover the effect of vinegar on the digestion of starch. Include experimental details in your answer and write in full sentences. If you've been following me for a while, you'll know that I prefer the variable layout as opposed to columns. So I'm going to use that today. The independent variable is what you change. So it's going to be the concentration of vinegar applied, and give some examples. So we're changing the concentration of vinegar. The dependent variable, which is what we measure. So we're looking at the digestion of starch, but it has to be something tangible that we're measuring. So I'm going to look at time taken for iodine, because remember iodine is used to test for starch to turn brown. Because when it turns brown, I know that starch must have been digested because it's no longer blue-black, the iodine. Control variables. So what am I keeping the same? So for sure, temperature, volume of starch solution, concentration of amylase, volume of iodine, and then lastly just state, repeat the experiment and calculate an average. I'll just show you where the marking points are awarded. So there's a mark for pointing out that you're using different concentrations of vinegar. So that's 1 mark. There's a mark for using iodine. And then there's a further mark for saying about how long it takes for that iodine to turn brown. Control variables, you get a mark for temperature and volume of starch solution. There's actually only 2 marks for control variables, and then repeat and calculate an average. So I've got 1, 2, 3, 4, 5, 6. So I'm happy that I've got my 6 marks.